Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your biochemistry mentor. Well, in today's video we are going to discuss a very an interesting and an important topic. In every video of mine I say interesting and important topic because all topics are important, isn't it? So today we will be discussing about the oxidation of fatty acids. Now see, what is a fatty acid? Okay, because whenever we talk about biochemistry, we should know that biochemistry is what? It is the chemistry of all the biochemical reactions which are going in our body. So these fatty acids are simply the compounds which have a hydrocarbon at one end and a carboxylic group on the other end. Means they have a hydrophobic as well as a hydrophilic areas, right? Now these fatty acids, when they combine with glycerol, they are going to form triacylglycerols, or we can call them as TGs. So how do we define TGs? TGs are the triesters of fatty acid and glycerol. Now simple concept is, whenever our body is in well-fed state, when we are eating something, well-fed state may always remember there will be an anabolic reaction or synthesis. So I can say well-fed state may synthesis of fat will be there. Fat or synthesis of TGs will take place, which is known as lipogenesis. All the time we cannot stay in a well-fed state, okay? Because we are having our breakfast, then uske baad we are giving a 4-5 hours ka gap, then we are having our lunch, again giving a gap of 4-5 hours, having the last meal, and again after a gap of 10-12 hours we will have our breakfast in the morning. What happens if we skip? So our body undergoes different phases. So well-fed ke baad it will be early fasting, it will be delayed fasting, prolonged fasting is starvation, right? Prolonged starvation is when our body is left over with none of the foodstuffs. Again our body manages to survive. So this is what is the beauty of the biochemical reactions. So well-fed state mein lipogenesis will take place. So if I talk about fasting, so fasting is say some 6 to 12 hours. Okay, it can go to 6 to 16 hours also. Then the only process which will give energy would be glycogenolysis. Right? You can say 6 to 12 hours, it can be 48 hours also. We will keep it now. Because fasting maybe I am just writing it like this because fasting maybe there are different stages. Early fasting is there, you can say late fasting is there, delayed fasting is there. So I can say that prolonged fasting is say 16 to 48 hours. Then what is happening here is the body gets energy from gluconeogenesis. The body gets energy from fatty acid oxidation. Right. And always remember, fatty acid oxidation produces acetyl coenzyme A. And this acetyl coenzyme A promotes gluconeogenesis. Right. And we can say another process is ketone body synthesis or ketogenesis. And if we talk about more than 2 days, it is like starvation. again body will undergo ketone genesis or lysis, and obviously proteolysis also. So this is how the body cycle goes on. So always remember, whenever we talk about any process which is occurring in a well-fed state, we call it an anabolic process. And whenever a process is involving the breakdown of something, we will call it as a catabolic process. So I can simply say that genesis, as it is clear from the word itself, genesis is a anabolic process. Glycogenolysis is a catabolic process. Fatty acid oxidation is a catabolic process, right? And that is what is our topic all about. It is the oxidation of fatty acids. So obviously, if oxidation of fatty acids is there, one can easily come to this conclusion that it is a catabolic process. Very important. Now, it is a catabolic process because breakdown is there and it will give energy. On the other hand, if it is catabolic, what is the site? The site has to be mitochondria, because whenever we talk about any reaction which is basically, which involves oxidation. And why mitochondria? Because mitochondria are the powerhouses of the cell. So something related, whenever we talk about the energy, whenever we talk about breakdown of something, or whenever we use the term oxidation, we always confine it to the mitochondria. Now here one thing very important is, if we talk about glycogenolysis, does not require oxygen. It is simple breakdown, anaerobic breakdown of glycogen to form glucose, because it is occurring in the skeletal muscle. This is very important, and that is why glucose stores glycogen, okay, and then excess amount of glucose is converted to form fat. Initially, we get energy from the breakdown of glycogen because it does not require oxygen. But fatty acid, no doubt they are the instantaneous sources of energy, but their oxidation is there, so it is confined to the mitochondria. So my site is mitochondria, or we can say mitochondrial matrix, right? And if we talk about where it occurs, so it can occur in the liver, it can occur in the muscle, it can occur in the adipose tissue. Right. Now catabolic hai, so we can say when it will occur during fasting stage. So these are the three very, very important points to remember about the oxidation of fatty acids. One is, it is a catabolic process. Since it is catabolic, it will occur in the mitochondrial matrix of the liver, of the muscle and the adipose tissue. Since it is giving energy to the body, occurs in the fasting stage. Right. Then if we talk about the TGs, we know that these triglycerides will be broken by the enzyme, you can say hormone sensitive lipase, or I will simply write lipase here in general. Basically it is HSL. Okay. So these TGs will give fatty acids and glycerol. Now the question is, we have already studied about the nomenclature of these fatty acids in detail in our video on chemistry of the fatty acids. So if you want to go into detail, if you want to know the details of this, you can just go to that. Here what you have to remember is that fatty acids can be even chain and odd chain. Even chain means like 2, 4, 6, 8, 10, 12, 16, 18 carbon atoms, and odd chain means 3, 5, 7, 9 carbon atoms. So even chain fatty acids always results in the formation of acetyl coenzyme A. Right, and odd chain fatty acids always results in the formation of propionyl coenzyme A. Right, fatty acids, if they are even chain, they will always form acetyl coenzyme A. If they are odd chain, they will always form propionyl coenzyme A. This acetyl coenzyme A undergoes TCA cycle. Right. So obviously 10 molecules of ATP, or if we talk about 2 acetyl coenzyme A, depending upon how many acetyl coenzyme A are liberated. And this propionyl coenzyme A undergoes the formation of glucose by forming succinyl coenzyme A, or you can say glucose, because it itself is an intermediate of TCA cycle. And that is why we say propionyl coenzyme A also undergoes gluconeogenesis. So propionyl coenzyme A forms succinyl coenzyme A, which is the intermediate of TCA cycle, which will be converted into glucose. Right. So now what happens here is, what we are going to study here is that how this pathway takes place, what is the energetics of this, and what is the regulation of this. And if we talk about glycerol, we know that glycerol also enters glycolysis, or you can say gluconeogenesis, like this. Clear? But this reaction does not take place in the adipose tissue because of the absence of glycerokinase enzyme. So now coming over to this, now what we are going to study here, that if we talk about a fatty acid, oxidation, there are some major pathways and there are some minor pathways. So if we talk about the major pathway, and if we talk about the minor pathway, ठीक है, major pathways will include only beta oxidation. Major pathways of oxidation of fatty acid includes beta oxidation, and minor pathway includes alpha oxidation and omega oxidation. Right. Now coming over to this, what is this beta, alpha or omega? So if I talk about in general the structure of a fatty acid, 1, 2, 3, 4, 5, 6, or for example this. Okay. So this is your alpha carbon atom adjacent to the functional group. This is beta carbon atom. Okay. So if oxidation is taking place here, this is alpha oxidation. And if oxidation is taking place here, it is beta oxidation. And if we talk about say R, say this is the omega carbon atom, say CH3 is there. If it is here, it becomes omega oxidation. Now, whether it is omega, whether it is alpha, the major pathway of oxidation is beta oxidation, like this. Very, very important. Okay. So we can say that the major pathway of oxidation of fatty acid is beta oxidation, which is taking place in the mitochondrial matrix. So now, what are the steps for beta oxidation? For beta oxidation, first of all, we have to activate the fatty acid. So first step is activation of fatty acid. Now, why activation is required? Because fatty acid is having COOH group, and we know that COOH group hai, so it is polar. Okay, so to make it non-polar, we activate the fatty acid, and obviously activation will require ATP. Okay, so first step is activation of fatty acid. Second is, just now I told you that beta oxidation is taking place where? In the mitochondria. And lipogenesis or the synthesis of fats is taking place in the cytoplasm. So, whenever fatty acid has to undergo oxidation, it has to be transported from the cytoplasm to the mitochondria. So, second is transport of fatty acid from the cytoplasm to the mitochondria. And once the fatty acid reaches the mitochondria, third step is it will undergo oxidation. So, third step is simple oxidation will be there, which takes place in 4 steps. Now, coming over to this, how activation takes place, how it is transported, and how this oxidation takes place finally in the end, how it is regulated, what is the energetics, that is all what we are going to discuss now. So, first step is activation. Now, whenever we have to activate a fatty acid, energy is required. And energy kahan se aati hai? It always comes from the energy currency or of the body, which is ATP. So, what will happen here is this fatty acid will be converted into acyl coenzyme A. Basically, what we are doing is this fatty acid C double bond OH is being converted into SCOA. SCOA. Why SCOA? Because SCOA is nothing but it is the precursor of vitamin B5, which is pantothenic acid. And here the name of the enzyme is acyl coenzyme A synthetase. And we know whenever we write synthetase, that means ATP is utilized. AMP, so I can say 2-phosphate are used. So, fatty acid is converted, or if you can say polar form of fatty acid is converted into the non-polar form of acyl coenzyme A in a simplest language. Okay, and this reaction is taking place in the cytoplasm. This is very important. Because fatty acid ki synthesis takes place in the cytoplasm, so obviously it has to be in the cytoplasm. Then second step is the transport. Transport of the fatty acid. Now we can say that transport of acyl coenzyme A, it is no longer a fatty acid. So I can say transport of acyl coenzyme A to the mitochondria. Now, if we talk about this step, first of all, you should know what is the difference between acyl coenzyme A and acetyl coenzyme A, because most of the students have confusion in this. So, always remember, if we are writing acetyl-CoA, okay, number of carbon atoms are only 2. So, it is a 2 carbon species, right? Or it is this CH3 C double bond O SCOA. But if we are writing acyl coenzyme A, it can have any number of carbon atoms except 0, any number. And if we have to write the structure, we can write the structure like this, C double bond O SCOA. So you have to understand the difference between acetyl coenzyme A and acyl coenzyme A. Okay, now see how it is transported. ऐसा तो नहीं है कि acyl coenzyme A will just walk from the cytoplasm to the mitochondria, it requires a shuttle. Shuttle is like, जैसे we also take a shuttle bus service from, if we have to move from one place to another. Same is the case here. It will take a kind of shuttle, and that shuttle is known as Carnitine shuttle. Very, very important. Now this Carnitine shuttle, basically what is Carnitine? This Carnitine is derived from lysine. And this is one of the very important significance of lysine also, because lysine is an essential amino acid, if you remember. And lysine is a ketogenic amino acid too. So derived from lysine, and this reaction requires vitamin C. Right. And if we talk about carnitine, carnitine is simply beta hydroxy gamma methyl ammonium butyrate. Right. So carnitine shuttle is, which is derived from lysine in the presence of vitamin C, is required for the transport of this acyl coenzyme A into the matrix of mitochondria. Now how this reaction, how this shuttle system is operative? This is again a very interesting process. See, there are two enzymes basically. This is the outer mitochondrial membrane, right, and this is the inner mitochondrial membrane. Acyl coenzyme A is here because this reaction is taking place in the cytoplasm. This is the intermembrane space. Okay, so here what happens is this acyl coenzyme A will combine with carnitine. Very important. So reaction will take place here, acyl coenzyme A plus carnitine. And the enzyme present here is CPT1, or we can call it as CAT1. If we are talking about acyl coenzyme A, the name of the enzyme is carnitine acyl transferase 1. Very important. Okay. And if we talk about CPT, so it is carnitine palmitoyl transferase 1. So I can say if it is palmitic acid, so we can simply say palmitoyl-CoA here. So the enzyme will become CPT1, carnitine palmitoyl transferase 1. Now what happens is this acyl coenzyme A plus carnitine, they are going to form what? They are going to form acyl carnitine. And that is what is our motto. And this acyl carnitine will come here, releasing carnitine and acetyl-CoA. And this carnitine by translocase will go out like this. And we can say that we get acyl coenzyme A. Right? And here the enzyme is CPT2 or CAT. Okay. So I'll just repeat it once again. See, if we talk about the outer mitochondrial membrane, CPT1 or CAT1 is present. If we talk about the inner mitochondrial membrane, CPT2 or CAT2 is present. What is the function of CPT1? CPT1 will let the formation of acyl coenzyme A plus carnitine to form acyl carnitine. Once acyl carnitine is produced, it will enter into the matrix of mitochondria and inner mitochondrial membrane pe CAT2 is present. Carnitine acyl transferase 2. Now we get this acyl coenzyme A. It has already reached there. Now this carnitine will further go out to react with more and more of acyl coenzyme A. So actually this acyl coenzyme A will now undergo beta oxidation. And this carnitine by translocase enzyme, this carnitine by translocase will go out to bring more and more of acyl coenzyme A, right. So this is your second step. Carnitine shuttle is required for the transport of acyl coenzyme A from the cytoplasm to the mitochondria, and the most important point which you have to remember here is CPT1 or CAT1. It is one and the same thing. It is present in the outer mitochondrial membrane. Present in the outer mitochondrial membrane. And if we talk about CPT2, carnitine palmitoyl transferase 2 or CAT2, it is present in the inner mitochondrial membrane. And the reaction takes place in the intermembrane space. Then we come over to the final step, which is the third step. Third step is beta oxidation. Now, whenever we talk about beta oxidation, it takes place in 4 steps. Takes place in 4 steps. First step is oxidation. Right. Second step is addition of water, which is hydration. Third step is again oxidation. And fourth step is cleavage, and the cleavage here is thiolytic cleavage. Very, very important. Right. So beta oxidation overall takes place in 4 steps: oxidation, addition of water, oxidation, and in the end thiolytic cleavage is there, right. So, and just now I have told you beta oxidation hoti kahan pe? It occurs on the beta carbon atom. So if I talk about, say, if this is a fatty acid, for example, CH2, CH2, and this is, say, C-double-bond-O, 𝗲𝗲𝗃. This is our acyl coenzyme A. Now fatty acid is not fatty acid. It has become acyl coenzyme A. I am not writing acetyl coenzyme A. I am just writing acyl coenzyme A. Why? Because it has number of carbon atoms, not 2. So this is your alpha carbon atom. This is your beta carbon atom. So oxidation will take place here. This is the site of oxidation. And you see, after 4 steps, this particular thing will be released. That is what we are going to study. Now, I told you the first step is oxidation. So, FAD is converted into FADH2. And what kind of product is produced? Are CH2, CH2, CH double bond CH, C double bond O, SCOA. It is going to pick one hydrogen from here, one hydrogen from here, right? And this is the first step, oxidation. So we can say that this is alpha beta. So alpha beta unsaturated acyl-CoA. right? Now, second step is water addition or hydration. So, we can say water is added, water is HOH. So, it will be like RCH2, CH2, CHOH and CH2, C double bond O, SCOA. So, this becomes alpha beta. So, I can say beta hydroxy-acyl coenzyme A. If we look at the third step is again oxidation. Oxidation. Here the acceptor is NAD. So, NAD will be converted into NADH plus H positive and these two hydrogen will be given out. So, we can say the next step is RCH2, CH2, C double bond O, CH2, C double bond O, SCOA. So, again we can say this is beta keto-acyl-CoA. Now, here you can see beta everything is occurring on the beta carbon atom, whether hydroxyl group is there, whether you can say keto group is there. So, it will form beta keto-acyl coenzyme A. Then we come over to the last step. Last step is cleavage. And the cleavage, I have mentioned this terminology, thiolytic cleavage is there. Now, what do you mean by thiolytic cleavage? Thiolysis means another coenzyme A is required here. And why this coenzyme A is required? Because the splitting will now take place here. And what we will get? If you see the products, we are going to get CH3, C double bond O, SCOA plus what is the remaining over? It is CH2, CH2, C double bond O, SCOA. Right. And if we have to name them, this is acetyl coenzyme A and this is acyl coenzyme A with 2 carbon atoms less. Carbon atoms less than the parent carbon atom. Right. And this is how the cycle will keep on going on unless and until we get either acetyl coenzyme A or we get propenyl coenzyme A plus acetyl coenzyme A, because if even chain fatty acids are there, obviously the end product has to be even carbon atoms. But odd chain fatty acids are there, the last molecule has to be a odd chain and rest it will be acetyl coenzyme A only. Okay, so this is how the process is of beta oxidation takes place. So, I can simply say that first is this is our acyl coenzyme A. Since oxidation is occurring at the beta carbon atom, first is oxidation, so it is going to form alpha beta unsaturated acyl-CoA. Then hydration is there, so beta hydroxy-acyl coenzyme A. Then again oxidation, so beta keto-acyl coenzyme A, and in the end acyl coenzyme A and acetyl coenzyme A are produced. Now the question is how to remember this. Okay, so there is one very easy formula to remember the number of cycles, and the formula is, if we have to calculate the number of cycles, it is n by 2 minus 1. What is n? N is number of carbon atoms. Okay, now if we say for example palmitic acid is there, because in our body mainly palmitoyl esters are there, so palmitic acid has 16 carbon atoms, right? So if we talk about 16 carbon atoms, I can simply say 16 by 2 minus 1. So 2 8s are 16 or 7 cycles. right? And second is how many acetyl coenzyme A are produced, because you can see 16 carbon atoms bane, palmitic acid is a even chain fatty acid. So number of acetyl coenzyme A or number of acetyl coenzyme A produced would be N by 2. So it is 16 by 2 equivalent to 8 acetyl coenzyme A. So I can simply write it down like this, that one molecule of palmitic acid which has 16 carbon atoms will undergo 7 cycles of beta oxidation leading to 8 molecules of acetyl coenzyme A. This you have to understand. One molecule of palmitic acid having 16 carbon atoms will produce 7 cycles of beta oxidation producing 8 molecules of acetyl coenzyme A. Now the question is, what is the energetics? Because so much is occurring, we are oxidizing palmitic acid. Palmitic acid has to travel from the cytoplasm to the mitochondria. So there must be some significance behind that, or it must be because fatty acids, I told you that they are the instantaneous sources of energy, and during fasting, after gluconeogenesis, they are the ones which are actually forming or giving energy to the body. So what is the energetics of this now? Right? So energetics of beta oxidation. Now we will just follow palmitic acid only because we have given the example of palmitic acid. So palmitate, which has 16 carbon atoms. Now it is undergoing 7 cycles. Up 7 cycles means 7 into FADH₂, because 7 FADH₂ will be produced. Just now you saw in the first step. And if we talk about NAD, again 7 NADH will be produced. And if we talk about acetyl coenzyme A, 8 acetyl coenzyme A will be produced. Okay. Now if we talk about the hydrolysis, we know that one molecule of FAD on hydrolysis gives 1.5 ATP. One molecule of NAD gives 2.5 ATP. So what will happen is 7 into 1.5, 7 into 2.5, right. And if we talk about acetyl coenzyme A, one molecule of acetyl coenzyme A gives 10 ATP via TCA. So 8 will give 8 into 10. So if we calculate it, it becomes 10.5, 17.5 and 80.0. So it comes out to be 10, 8, 108.0. So we can say that one molecule of palmitic acid will give 108 ATP molecules. Now go back to the history. The first step. First step में क्या था? First step was कि 2 ATP मतलब 1 ATP equivalent to 2 phosphate were used. So here that also has to be taken into consideration because that was an activation step. So we can say that ATP used for activation so it will be minus 2. So it comes out to be 106.0 ATP. So always remember, one molecule of palmitate via beta oxidation will give 106 ATP molecules. This we have to remember. And this is very, very important, that one molecule of palmitic acid via beta oxidation will undergo 7 cycles producing 8 acetyl coenzyme A, forming 1 not 6 ATP molecules. So if we know the number of carbon atoms, we can actually give the solution that what will be the energetics. Just a second example, if I tell you, second example is stearic acid. Now stearic acid has 18 carbon atoms. Okay, just apply the formula, n by 2 minus 1, so 18 by 2 minus 1 is equal to 8 cycles, and n by 2 is equal to 18, 9 acetyl coenzyme A. Right. So I can say that 8 FADH₂, 8 NADH plus H positive, and 9 acetyl coenzyme A. Okay, just calculate it. It will be 8 into 1.5, 8 into 2.5, and 9 into 10. So it comes out to be 12.5, 20.5 and 90.0. 5, 5, 10, 3, 9, 10, 11, 12, 13, 133.0, or if we say minus 2 ATP which are utilized for activation, so it comes out to be 130 ATP molecules. 131, 3 minus 2 is 1. Okay, so this is the energetics of stearic acid, and this is what is beta oxidation. Now, just now we have studied the beta oxidation of even chain fatty acids. So you can say that beta oxidation of even chain fatty acids will obviously form acetyl coenzyme A. But if we talk about the beta oxidation of odd chain fatty acids, they will produce one molecule of propanol coenzyme A plus acetyl coenzyme A. Acetyl coenzyme A we can predict kitne banenge depending upon the number of carbon atoms, but always remember end will be with one molecule of propanol coenzyme A. Because suppose if I take the example like this, 1, 2, 3, 4, 5, 6, 7. If this is the case, okay, this is alpha, this is beta, so I can say that यहाँ पे C double bond O SCOA, this is alpha, this is beta. So we can say one oxidation will be here. Okay, so ये क्या बना देगा? This is going to form this type of structure, SCOA. Then again this is alpha, this is beta. So if oxidation is here, so it is going to form this type of compound, SCOA plus CH3 C double bond O SCOA. So this is having 3 carbon atoms, so this will be propanol coenzyme A. So this is one example I have quoted. This is 1, 2, 3, 4, 5, 6, 7. So I can say that when one molecule of heptanoic acid means it has 7 carbon atoms, so it is an odd chain fatty acid. So heptanoic acid is going to form 1 or 2 acetyl coenzyme A and 1 propanol coenzyme A. Clear? So now after oxidation, the main part is regulation. Now when we talk about the regulation, always remember this is lipolysis, this is a catabolic process, so always keep one thing in mind, that insulin will inhibit this, because insulin is an anabolic hormone and glucagon will promote it. Similarly, epinephrine Similarly, thyroid hormones, they are going to promote it. This thing we have to keep it in mind. Now the question is that glucagon, epinephrine and thyroid hormones, if they are accelerating it, if it is inhibiting it, so what is the main target? So here the main target is CPT1 or CAT1. And that is why CPT1 is the rate limiting enzyme of beta oxidation. So rate limiting enzyme is carnitine palmitoyl transferase 1 or CAT1, because this is the one which is actually helping the entry of the fatty acid for oxidation. Okay, and again there is one more enzyme which we will be discussing in detail in the lipogenesis, acetyl-coenzyme carboxylase. Okay, this is a rate limiting enzyme of lipogenesis. So there is a kind of coordination between them. So acetyl-coenzyme A, if it is coordinating lipogenesis, CPT will be coordinating lipolysis, and the main enzyme here is hormone sensitive lipase, which is also known as HSL. So insulin inhibits HSL, whereas these promote HSL, which is hormone sensitive lipase. Right. So we can simply say that कि beta oxidation of fatty acids में the rate limiting enzyme is CPT1 or CAT1, which is regulating the flux of this acyl coenzyme A into the mitochondria. So abhi just now we have discussed about the beta oxidation of, because if you go back again to the chemistry, I told you that as per the carbon atoms, there are different type of fatty acids like SCFAs are there, medium chain fatty acids are there, long chain fatty acids are there, and very long chain fatty acids are there. Okay, the basic difference between them is that SCFAs and MCFAs do not require carnitine. Do not require carnitine for the transport. Long chain fatty acids occur in the mitochondria, which we studied now, and yes, they require carnitine. And VLCFAs mainly will take place in the peroxisomes and mitochondria. Okay, so in this video we have discussed just up till this segment. In the next video we will be discussing about the beta oxidation of very long chain fatty acids which occurs in the peroxisomes or the mitochondria, and we are going to study the minor pathways also, alpha oxidation and omega oxidation. So this video is exclusively on the beta oxidation of the odd chain and even chain fatty acids, which produces acetyl-coenzyme A, and odd chain produces propenyl coenzyme A and acetyl coenzyme A, which is occurring in the mitochondrial matrix requiring carnitine shuttle. So I hope this video is clear to all of you. Thank you so much.